So my plan is to finish up with visual pathways if I possibly can, so that we can start object recognition on Friday and get as far as that as we can, and still review on Monday. But so we were just talking about the so we had gotten information to the visual cortex. It went to 2 separate pathways from there for higher level processing, to the parietal lobes, the where-how pathway and that dorsal route, and to the temporal lobes, that what pathway, or the object recognition pathway down the ventral route. And so I actually had just given some examples of some of these neurons that are beyond feature detectors. So we've gotten a little bit more complicated, and then eventually we got to faces and the fusiform face area, that module of processing. In addition to the fusiform face area, we also have a couple other areas that have been researched quite a bit. We are finding more and more of these areas, but so the perihippocampal place area is another area that's been examined, again, quite a bit. So if we look at the top row, or if the participant is looking at the top row of pictures there by the PPA one, so whether it's outdoor scenes or a picture of a house or an empty space that is the corner of a room, or even what looks to me like kind of like not like Lego, but yeah, like little Lego houses, something that suggests navigation, or that we would need to remember for how to navigate around. The perihippocampal place area is active, and you can see on the left-hand side that we're getting really good spatial resolution and being able to see exactly where this perihippocampal place area is. Meanwhile, if we show it other objects, whether it is like the jet plane or a jumble, or I'm not really actually sure what those other things are, the single Lego piece that's all mixed up, that's not giving me kind of spatial information that might be used for navigation, then we do not see that area as active. The extrastriate body area is another region that has been discovered and examined a bit. So if we are looking at the top row next to the EBA, extrastriate body area, whether we're looking at a person's arm or a whole person kind of curled up, a stick person, a shadow person, this area, and you can see again to the left the picture of the brain, the fMRI response. We can see pretty specifically where we have more activation there to those kind of body regions than to tools or jumbles, or I don't know what those little letter-like things are that are all mixed up, or faces. So I saved faces because remember we have a fusiform face area, so that's not included as part of the body area. And while these modules are really neat and really interesting, that we see a particular category really being processed and more activity in that region to that category than to other categories, remember that we still have a very distributed representation. There's still activation in other areas. Object identification causes activity that's distributed over a wide area of the brain, starting with the visual cortex and into many of these, not just the temporal lobe regions for object recognition, but as you can see in the pictures, into the parietal lobes as well. The picture on the far left is looking at the segregation by category, so really where a particular category had its maximal response. And so houses you can kind of see over on the right side, and kind of up towards the perihippocampal, so up towards the parietal. Chairs, lower left, but we see other little areas, but a bit of it, quite a bit, is on the lower left. Faces, you can see the strongest activation is, as I said, with the fusiform face area and the infratemporal cortex down there on the right side, but you can see the left side somewhat activated, and then the white spot is where there's no real difference between those categories. But if we pull that out and we're not looking at the maximal response, we're just looking at response magnitude, usually, again, with some subtraction, some task that's been subtracted out, but where we're looking at response magnitude, right? You can see, and I'm gonna pick faces, so I'm not going to talk about all these, but if you look at the middle one, fusiform face, the one where the participants are looking at faces, that there is, so the green and blue are pretty low activity, red, orange is getting into higher activity, and yellow is my highest activity. And you can see the highest activity is right there in that fusiform face area. But again, now we see a very distributed representation when we're just looking at, when we're not doing that comparison and saying maximal response compared to others, when we're just looking at what's the response to faces, we see a very distributed neural firing in many areas of the brain. So for a long time after we realized that we had these modules and they were processing specific categories of objects, what most people used for stimuli were still pictures, like what we've been looking at. But in 2012, Huth et al. Did this same kind of study where they looked at short movie clips and they labeled those movie clips to see what was in it. So I'm going to pick the bottom right one because it has kind of more different stuff in it than some of them. So an animal to bison, the verb, the action is walking, has grass and a stream in it. So they would label those movie clips, and then what you can see on the right is the brain activity to some of those objects and some of those actions. Just for example, you can see the kind of yellowy color is representing where people are processing information about animals, and that we do have a couple of places in the brain that are active. And then I have talking, or I see talking in a couple of different areas as well. Maybe one up towards, I'm assuming that's Broca's area, where we have this kind of, what do you call it, embodied action that we move we don't move our lips necessarily, but our Broca's area is active as if we're moving our lips when someone else is talking. So just again, very distributed and now a bit more naturalistic of a stimulus. And I'm going to pound this message home with one more example because I like this example. But when we're responding to a person's face, we're really responding to various aspects of that face at the same time, as I'm sure you can imagine. That so there are emotional aspects, both of how the person is looking, their own emotion, as well as our own emotional response to that person, where we see activity in the amygdala, at the very least, at least the amygdala. We look at where somebody is looking and how parts of the face are moving. So if we take this example of somebody talking about something and they're looking off, we might think that where they're looking to has something to do with what they're talking about. So we're gonna look at where they're looking. If we really want to understand them, we might look at how their mouth is moving. We might look at whether or not they're smiling and their face is moving kind of in different emotional ways. So and in that we see at the very least, again, at least the structure of the superior temporal sulcus, which tells us about some of the social processing and biological motion. We judge how attractive a face is, and your author puts this as what's going on in the prefrontal cortex or in the frontal lobes. You can kind of see that in the picture there. I like to put a bit more in my frontal lobes. I like to think that maybe I'm thinking about whether I want to work with this person or what kind of research this person does, this sort of more evaluative aspects of a person. And then whether or not the face is familiar. So the amygdala, as well as other areas, the facial recognition in the fusiform face area, memory areas, the initial visual processing in the occipital lobe, primary visual cortex. And so, again, very distributed representation. And here is the author walking you through everything I just said with mostly the same stuff. So if you want to pause on the table. Okay, so this is a good place to talk about how the firing of neurons is actually representing various characteristics of the environment. One possible explanation is called specificity coding, where the representation of some particular object in the environment is represented by the firing of a specific neuron that is tuned to fire to that object. These are sometimes referred to as grandmother cells, a neuron that responds only to a specific stimulus. So I have a cell that responds to my grandmother is the idea. And this can be an image of my grandmother's face, the concept of grandmothers, maybe my grandmother's name, anything that gets tied to that's really grandmother-like. To some extent, there was research that by Quiroga and colleagues where they found, I think in the hippocampus, certain neurons that would respond to, for example, Halle Berry, where they were looking at people who were about to undergo surgery for epilepsy, and they sort of had this exposed, and they were recording, and they were showing pictures of different stars and different famous people. And this neuron that responded to Halle Berry wasn't responding to other pictures that it was shown, and it responded to a picture of Halle Berry in a regular dress, in her Catwoman costume. It responded to Halle Berry's name. So this was really where they got the idea of this possibility that there are these grandmother cells. Now I'm going to throw a monkey wrench into that in a minute, but I'm going to continue walking us through this possible explanation for how the nervous system is really representing objects in the environment. So I'm hoping that was really clear, but I do like to talk from pictures a bit. So just to give you what I think he's changed slightly in the book, but it has the same basic idea. So the idea of specificity coding is I have a neuron, let's say neuron 1, and it responds to Bill. I have a different neuron, neuron 2, it responds to Mary. Neuron 1 does not respond to Mary, and neuron 3 does not respond to Mary. Neuron 3 responds to Raphael, and it does not respond to Bill or Mary. So that's really the specificity coding in a picture form. And I usually pause and take a minute and ask the class here, do these neurons really exist? And really, why is it unreasonable for our entire nervous system to work this way, for this to be the way that we code the objects in our world? And while you might want to pause again and think for a little while longer, I'm going to give you, so students come up with a number of answers, and some of the ones that always strike me as most memorable, and I do this kind of, yeah, of course, so exactly. One, so as we get older and we keep meeting new people, this would mean that we'd either have to have neurons that were not responding to anything and are now being utilized for this particular person, and we know that's really not how neurons work, or we have to create new neurons. And this isn't a place where, well, the hippocampus is a place where we're creating new neurons, but not in this way where we're just continuing to create new neurons and new neurons, or our hippocampus would grow and grow, right? It just doesn't, it's not the way. It doesn't make sense for really learning new people, meeting new people, and getting to know them. I used to, when we had face-to-face classes, I got to know the names and faces of about 200 people a semester. So I'm constantly adding those people, and that just doesn't make sense for the nervous system to code for them that way. I'm trying to think of another one. There are several reasons, and they're not coming to my head right off today, but there are several reasons why this is really unreasonable as a way for the nervous system to work. Not to mention the fact that we know that there is distributed representation. So what is this about a particular neuron that's going to, like one specific neuron that represents a specific person? So if we aren't using specificity coding to recognize a person's face, what are our options? Well, one is distributed coding. And this is saying something somewhat different than, yes, processing is distributed around the brain because we are responding to all these different aspects of the face. This is a question of what neurons, or what is in the nervous system that is telling me this is the object that is in my environment. This is the face that I am looking at. And distributed coding basically says that we represent a particular object or a face by the pattern of firing in groups of neurons, and so larger groups of neurons. Sometimes this is called population coding, as they talk about it as populations of neurons. This doesn't require so many specialized neurons, so there's not this, well, every time I learn something, I have to add another neuron. It starts to feel like that would have to be an infinite number of neurons just in my hippocampus and temporal lobes to remember everything. Another option, however, from specificity coding and distributed coding is sparse coding, where a stimulus is represented by a small number of neurons and their pattern of firing. And this, in the case of recognizing faces and these kinds of concepts, this appears to be how the nervous system is representing things. Now, we're going to come back to this question of specificity coding versus distributed coding in various sensory modalities. But for this question, if we look at those Quiroga et al. Results, I mentioned Halle Berry before, but Jennifer Aniston was another one of those faces that they showed. I think the one in the book is, I can't say his name, so why am I saying it? Stephen Carell? Anyway, what they were saying was they were recording from certain neurons, and so for them to have picked the faces that would cause those neurons to respond would be like looking for a needle in a haystack when you're just sticking the needle up into a haystack, or you're sticking your electrode up into a haystack and showing pictures. So if it was specificity coding, there's really almost no way they would have gotten neural response for all the famous faces that they did. Another thing they found, I thought was kind of interesting, was that this neuron that responded to Jennifer Aniston, it also responded to, Lisa Kudrow, so another person from that show Friends. So somehow they are semantically linked, and that neuron was responding really to both of them. So if you're really into your sports and you have a couple of people on a team that you know and really like, they might be kind of sharing a neuron. But again, it's going to be sharing that neuron amidst a small number of neurons that are firing, where there's a different pattern of firing for each of those people, if that makes sense. I hope so. If we take distributed coding and just give it a picture, so now neurons 1, 2, and 3, although again with distributed coding, we're going to say this is a large population, because to me this almost looks like sparse coding if I only have 3 neurons in my repertoire here. But we'll just distributed coding. I have neurons 1, 2, and 3, and it's the pattern of firing. So neuron 1 responds strongly to Bill. Neuron 2 and neuron 3 look more like spontaneous activity. 2 and 3 both respond strongly to Mary. 1 may be spontaneous activity. 1 and 3 respond to Raphael. 2 responds some, and so on. And we can add Roger, and there's now another pattern, different pattern of response. If we ask about how this neuronal specialization occurs, basically we know that to a large extent this is evolutionarily driven, right? We are born with the basic architecture of the brain. As it says here, the overall layout must have some preset basis. We actually are very similar in the structure and the functions of those structures to other animals, and especially as we get to primates, but mammals and so forth. However, we also know that to some extent there is experiential influence on neuronal specialization by personal experience. So the evidence is that the details are more shaped by experience. This is why somebody who is learning several languages, they're going to develop a much different part of their brain than I was talking in class today about people who play basketball. So people who play basketball have very large cerebellums. That's being that kind of timing and motor timing and and all of that, and timing socially with other people, that is all being developed a great deal when you're playing basketball. And one study that has really looked at this kind of neuronal specialization and how experience affects what neurons respond to is a study that was conducted by Isabelle Gauthier and her colleagues. They trained people. Oh, I always look this up and I didn't this time, but I think it's something like 7 hours and 4 days altogether. They trained people to recognize these, we're going to call them greebles, or sometimes they're called greeble people or greeble stimuli. And you can see on the left, these greebles are, they're kind of bird-like in their appearance. But they're different. They've got a couple beaks, and they're all, those four are all different from each other. All of the greebles in the study were different from each other, and they were all given their own names. So the people were learning to name these greebles. Okay, and we're going to look at the response using fMRI of the fusiform face area. And if we look before training, so before they've looked at these greebles and learned anything, that FFA area, so if we're looking over in the left set of figures here of the graph and the brains, that area is responding to faces and not responding at all pretty much to when they're being shown greebles. Okay, after this kind of extensive training, where yes, these people could name all of these various greebles, what we see is that area is responding to faces, and that area is responding to greebles. And so this is an example I have down there in the red. This is an example of experience-dependent plasticity, where we see a change in that neuronal specialization based on experience. And notice this is pretty short-term experience. And so the fusiform face area sometimes gets talked about as an expertise area. So people who memorize cars and know all kinds of cars, they have their fusiform face area responds to cars. Bird watchers who get to know all the different bunches of different kinds of birds, their FFA is going to respond to birds. So again, kind of an expertise area. I'm not really sure why we've never changed the name. It's been there a long time, I guess, and it's considered the fusiform face area, but it changes based on our experience. So we come back to our metaphysical question of how you feel about what you're learning and whether it feels like we're really explaining this hard problem of consciousness, how these physiological responses, the flowing of ions across a membrane or the releasing of neurotransmitters, are really causing or transforming into the richness of perceptual experience. So how do neural impulses become perception? Or does it kind of feel like we're still in this, well, we're addressing how physiological responses correlate with experience. And it seems more and more relatable as we get to recognizing objects, but it's still just correlations. It's something I think you should probably pause and think about a little. I will tell you, I often have students feel like we are answering the hard problem of consciousness, and I feel that I'm in this, we're still in the easy problem of really talking about correlations, but I don't know if some of that is a generational issue that you all have just been exposed for so much longer and so much younger to a lot of the neuroscience, that this field has blossomed and developed and become very sophisticated since I left graduate school. And when I went into graduate school, it wasn't necessary to learn a lot of, I didn't take any anatomy, I didn't take any neuroscience or brain and behavior, physiological psych, or and sensation and perception was not this heavy and geared towards the neuroscience. So yeah, I don't know how you all feel. I've also had classes say, yeah, we're nowhere close to the hard problem of consciousness. So think about what you think, how you feel about it. All right, I'm definitely out of time. I'm about, I think, 30 seconds over with my last one, and we will get to object recognition. Yay.